Yes, I'm Tim Johns. I work for the Environment Agency, and I'm based in Wallingford in Oxfordshire, so not too far away from Hampshire. And I'm what's called a groundwater ecology lead for the country. I say the lead, I'm actually the only one. It's a team of me. I've got a background in freshwater ecology, which is sort of my passion. Many years ago, when I started work, I started as a sort of groundwater monitoring sort of officer. And so I sort of married the two things together. Hence, groundwater ecology is really what I'm going to talk about. Right. So I think the first thing to do, the groundwater ecosystem or ground environment, I should say, is a unique ecosystem in many ways. It's obviously characterized by complete darkness. It's quite a strange environment. What's it does is characterized by is sort of reasonably low temperatures, which are sort of stable, don't get too much variation in terms of seasonal variation, annual variations, like you would do in a river, and dissolved oxygen concentrations again are lower than you would find in a river. Having said that, and also a rather important part is the fact that, of course, the groundwater ecosystem is there's no photosynthesis, sort of depends entirely on chemical energy sources, which are mainly derived from material that's percolated through the surface, from the surface, I should say, and they're consequently food chains are very sort of simple, simple structures. And then in terms of the physical habitat that we have the interstitial habitat, if you like, which is the sort of porous rocks where you've got little spaces between particles. Then we've got fracture networks and conduits and that sort of thing, which you get in certain types of rock. And then other types of rock, you get cave environments, which are often in the sort of karstic rocks, which we'll come on to talk to in a minute. Then really, I guess the next thing is sort of why is groundwater ecology important? I mean, historically, we've always just looked at groundwater quantity and quality in terms of chemistry and levels. So for water resources and water quality point of view, and so it's used for potable supply. So why is the groundwater ecology important? Well, I guess I'm talking to people who are interested in this thing. So the groundwater biodiversity, the actual species are intrinsically interesting, I think, and of conservation value. And in modern parlances, that we use natural capital, which is a slightly overused term, but anyway, we can come on to that in a bit. So that sort of drives me and my interest, I guess, because I've been doing this for some time prior to being that sort of groundwater lead, which I only sort of took up a year or so ago. I've been doing this sort of stuff back in sort of for about 15 years or more. So the other side, sorry, is the groundwater ecosystem functioning part. And what I mean by that is the sort of there's biogeochemical processes that are driven or facilitated by microbial communities in groundwater, and that they are grazed upon those communities, if you like, by these larger animals we have here, which graze upon these biofilms and help proliferate them, and they can drive ecosystem services, which we mean sort of attenuation of pollutions or pollutants rather, and water purification, if I can get that word out right. So basically a huge benefit to people and biodiversity. Without these things, services, we would struggle to live. Moving on to what lives in groundwater, perhaps unsurprisingly, the majority of the biomass and indeed the biodiversity in groundwater is actually microbial in nature, and of that, bacteria and protozoa are the main ones. But you do find fungi and viruses, for example, and you see there's a huge estimate of that globally. There are indeed also some rare amphibians and fish that you find in groundwater. Sadly, none in the UK. You may have come across if you've been to sort of northern Italy or Slovenia, where there's sort of the big karstic caves and the blind cave salamander called the Olm or Proteus is found there. And this is an ancient animal. It's been knocking around for 100,000 years or so. Hardly ever eats, and is a very interesting looking animal to see. So I do advise, if you go that way, go and have a look down one of those caves. There's big show caves where they're present. But anyway, I'm not talking about that. We're largely talking about invertebrate fauna. And as you can see in my slide there, the vast majority of these invertebrates are in groundwater are crustaceans. And the global estimates are a little bit they're a bit out of date, and I'm not sure how accurate they are. I don't think there is a really good accurate figure, but the one for Europe gives you an indication of how many obligate groundwater animals there are, crustaceans, I should say, in Europe. Now, turning attention there to the ground fauna, these macromyofauna. So it's called stygofauna or stygofauna. I say stygo because it's sort of slightly easier to say, and it means the word stygo comes from, or stygo comes from the river Styx, which is from the Greek underworld and the Hades and that sort of thing, which makes sense. So we'll stick with stygobites, but it might be pronounced the other way. So we've got stygobites, which are the sort of specialists, and they only live in groundwater. So the obligate groundwater animals. And then we have the generalists, which is the stygophiles, which can live in groundwater, but also live in surface water. And off the picture there, we've got an ostracod, little seed shrimp, as it's sometimes called, and a copepod. Now, they are generally stygophilic, but you do find stygobitic versions of those. And there's also another category which I haven't included, which is called the stygoxins, and they are basically accidental tourists turned up there in the groundwater, didn't mean to, then tend to last, either get out or die. Where do we find these little animals? So if we sort of make it quite simple, there's three sort of aquifer types that we could look at. We've got our sort of limestone and carbonate rocks, where we get solution features through water being dissolved in the carbonate rock, and so limestones and chalks I'm talking about here, and there's sort of castification, different degrees of castification in those rocks. So we have those, and so those pictures on the right, we've got fissures and fractures. Those two are from the chalk, and those sizes of those fractures can vary from sort of a few centimeters to several meters. And then, of course, in sort of bigger karstic situations, in some of the limestones, you've got caves, which you can indeed squeeze through, which that's obviously showing you can do. Then we got our fractured aquifers, so we got sort of sandstones and granites, which have been fracture networks have been developed through more tectonic movements. And then we have sort of unconsolidated material, which is valley gravels and that type of thing, which we find obviously in valleys, river valleys, but also there's this thing called the hyporheic zone, which is sort of a zone, is an ecotone, if you like, between the surface water and the groundwater on most rivers, so particularly groundwater fed rivers, essentially, or where there's a groundwater element to the flow. There's a sort of upwelling and downwelling, and then you have this environment within that hyporheic zone where it's different types of slightly different temperature to the surface water, etc., and the groundwater, and you get an interesting fauna within that environment. Okay, so that sort of covers the first sort of what the groundwater is about and where we're looking, etc. And now I thought we'd just have a look at the species and distribution we have, mainly in England and straying a little bit outside. So these are the species we find of those larger stygobites in groundwater. I've highlighted a couple at the bottom. So there's well, actually, there's two ostracods there. They've only recently been found, one in Scotland in a Scottish cave and one in the Mendips, I believe. And then we have a copepod, the Ankylocyclops sensitivitas, and that is found, or there's only one record for that, that's why I've highlighted it, and that was from 1933 or something. So I'm not entirely sure that's correct because we haven't found it since. And then the other two at the bottom, they're highlighted in yellow. The names Islandicus and Wextensis probably give you a clue that they're from Ireland, and though they're endemic species to Ireland, so we don't find those in England or Britain. And then on the right, we've got a diagram which is sort of indicating that some molecular work that was done a little while ago, and that actually continuing at quite a pace nowadays, and demonstrates actually within those species that we've got there, we've got a high phylogenetic diversity, which means that there's different species within those species groups, as it were. So they're slightly subtly different because they've been isolated, and therefore they're some distance relation to those European counterparts where they exist in Europe. So they may indeed be separate species. So there's more work to be done on that, but that's an interesting avenue. So I put this slide in because it's not very good. Not because I didn't put it in because it's not very good. I couldn't find a better one. And what I'm trying to show is just that actually, whilst to demonstrate in terms of the number of species that we have, so we've got a very limited number, but elsewhere in Europe, and this is say from 2009, this paper. So the numbers you can see in northern Italy and Slovenia there are high. So we've got up to sort of 79 or so species, of giving an indication of the numbers of species in the area. Elsewhere in the world, so that's a biodiversity hotspot for Stygobites, and equally in Australia particularly, there are absolutely hundreds of things, because you can imagine they've sort of lovely calcite rock, I believe, in the Pilbara in northwestern Australia, and elsewhere in Australia. So there's a which has been quite important because of their mining industry. So there's a lot of work being done there. So I just thought to sort of characterize what these animals look like. Obviously, they're not this big, and I put a range up to 15 mil. That's perhaps pushing it, and with where you might find them in caves, they can get a bit bigger, perhaps. But mostly they're quite small, and we'll look at those in a minute. So they're highly evolved for their environment they live in. Obviously, they don't need eyes, so they've lost their eye pigment, or eye development rather, and they've lost pigment, and so they're sort of pale or colourless and translucent sometimes. They're slender and elongated, sort of more worm-like, if you like, compared to, let's say, a gammarus, which is a freshwater shrimp, which is you're probably more familiar with, and that they've got these sort of long appendages to crawl through and that sort of thing. In terms of their length of life, they can live for up to 10 years or possibly. I kept one in the fridge at work for about 5 years, no, 6 years actually. So they can hang around for a while. They've got low reproduction rates. They all look the same, morphologically conversion, and they can tolerate those lower oxygen levels, which I talked about earlier. They're generally poor dispersers, so they've got these small ranges, but I mentioned those that's hyperaric zone, and a theory called hyperaric corridors, where they could potentially move in these shallow aquifers along those river gravel beds and move and perhaps get bigger ranges that way. But vast majority of them don't seem to move very far, and they're pretty much at the mercy of flow as well. Yeah, just looking at the different amphipod species. So those are listed. These were the main ones, and then we've got another couple of other species. The Niphagus fontanus and Niphagus aquex are the sort of the most common, or common in southern Britain. The Aquilex is probably the most commonly found, particularly in those riverine environments, whereas fontanus perhaps more cave-like environments. Of the other species, we'll have a look at the distribution in a bit, so I won't say too much other than the Niphagus cochianus is found very much associated with chalk, Niphagus glenii in the southwest of England, and Microniphagus is a very tiny little thing, and only recently found initially in Ireland in 2006, and then in England in 2010. And then of those other two species listed, we've got the isopod, which is very much like an ocellus or a water hoglouse, but as you can see, similarly again, elongated, thinner eyeless. And then we have this weird thing called the Anthrobathanella stammeri, which I did find once, and I even took a picture worse than that picture, and then I put it in the vial, so excited about it, came back to it next day, couldn't find it. So it was so small, I lost it. So I was a little bit disappointed, and I haven't found another one since. But as you see, that's the sort of thing they look like. I say we'll look at the ID a bit later. So moving on to the distribution, they've got a southern bias to the distribution you can see there, and that's probably in relation to the sort of glacial history of the country. As you can see, I've got a sort of purple line, which I drew on myself, so it's not very accurate. But it's basically the sort of roughly the maximal extent of the last Devonsian glaciation, which was about 33,000 years ago-ish. And then I think it receded about 15 to 12,000 years ago. And the idea is that the ice sheets covered these parts of the country, and they would lead to extinctions because they would have cut off the food supply, and also potentially oxygen levels would be lower. But as you can see, there's a few records north of that line. Now, those, interestingly enough, for that Microniphagus, which is this one here, and that's a record actually recorded quite recently. And these are this Anthrobathanella. Those are very small, so they could have moved through those hyperaric corridor type of things. So that they could have recolonized potentially that way. Oh yeah, and this other one up there is a very ancient record from early 1900s probably, and possibly incorrect Niphagus aquilex. So still, I'm a little looking. We may find something up there. As I said, it's very much in the chalk. Don't worry about the colours of the dots because we're going to come on to that in a minute. And then I should mention this last species, or this last bit, which is this Niphagus glenii, and that is an endemic, definitely an endemic species to Devon and Cornwall, and that's been knocking around about 19 million years ago, and possibly therefore one of the oldest, if not the oldest, endemic fauna to Britain. I stand to be corrected. Someone may know better than me about that. But what's interesting, we don't find it eastward, and that may be because of a mudstone sort of barrier that sort of goes through Somerset, Dorset sort of area on that border. It may turn up, and further, obviously, further monitoring may find it, but it's unlikely. So again, just look at their species, these map, the colours, I'm afraid, of the previous map don't match this. This was my error. But anyway, you can see the idea that the Niphagus aquilex is pretty well distributed, and I've included Wales here because the records include it. The other map was for another purpose. And so you can see aquilex actually is also beyond that Devonian glacial limit. No, many of those records are associated with caves, I should say, but certainly not exclusively. And then Niphagus fontanus, again cave related. Here, obviously Welsh sort of cave area, and the Mendips, but also found in the chalk. And similarly, you can see the line of the chalk in the Niphagus Cochianus ones. And then, as just said, Niphagus glenii very much in the southwest. Just to finish off the other ones. Niphagus liruthae, this Microniphagus is the tiny one. Weird sort of distribution, but since it's only been found recently, it's probably going to be found further. And say that a record is from Wales. I found another one just here, and I've been sampling up this way, so I'm hopeful I'll find more. Grangonyx, not very commonly found, but in the chalk and also in caves. Similarly, the Procellus Capaticus cave obviously gives a bit of a clue. And then finally, Anthrobathanella, we can see has quite an interesting distribution with a big gap in the middle here. Might be related to the geology a little bit, but also more likely to relate it to the fact that we haven't found it, it's been overlooked. And you can see there's one in Scotland, so clearly it's probably found much more widespread than we envisage. But when it's so small, it's either not seen or it's not detected. Turning our attention to the Hampshire area, the first thing that I guess to note is that the oldest records, or the records, go back to 1858, which is quite a while back, and this relates to an what's called the Old Town Well in Ringwood. Actually, lots of different species, not all found in 1856. So there's records there from into the earlier 1900s. But they found all these the species. So they found Procellus, Niphagus cochianus, Niphagus aquilex, and Grangonyx subterraneus. Unfortunately, that well seems to have disappeared, unless anyone knows where it is. The grid reference for it is so vague, it could be anywhere in that general sort of area. But anyway, so the next thing to note is all look at these number of records or sites where we found Niphagus aquilex, and with this you can see that these are largely related to the river network, and that again relates to that shallow hyporheic zone where potentially these things have been through upwelling, and they've come into the river and been captured that way. We'll look at that in a sec. Yeah, sorry, I should say the Isle of Wight, shouldn't I? So there's a few records on the Isle of Wight, and interestingly, we've one recent one, fairly recent, of Microniphagus. So that's quite good there. And then the other ones are again aquilex, mainly. I think I've said in the next slide where again I think these are rivers and actually in some shallow wells, and then one solitary Niphagus cochianus. This is a number of records that of positive records. I suppose we haven't got, unfortunately, a list of negative records, but positive records from since 1850 or earlier. As you can see, the sort of interest here. Then there was a bit of an interest in the sort of early part of the 1900s. It went quiet, war, and then 1950s or 60s, really, there was quite a bit of interest in groundwater fauna, and so that spawned quite a bit of sampling. And then that sort of actually died off again, and then resurrected itself in the sort of late 90s. A lot more sampling, and that reflects what's being done globally, actually. So interesting, the samples of the Niphagus aquilex, you can see There's most of the sources of the records are from rivers, and they're mainly aqueducts. So those all relate actually to environmental, or nearly always to environmentally kick samples in the river. So they've kicked quite hard, or there's upwelling, and these animals are being caught in there. And actually, the numbers you get in those river samples is actually far in excess of what you tend to get in the wells. I guess that's because they've been forced out to a degree through upwelling. And I think this just shows, again, the sort of numbers. And we can see with our sampling since 2000, we've nearly got all the species, but it would be really good to see if we could find Proceras, which hasn't been found since whenever it was in that town well in Ringwood. Back in 2021, Tim Sykes, who some of you will know, and my EA colleague, got me down to these sites of the Bourne Rivulet, etc. I think there'd been a BioBlitz, and I sort of was a bit late to the game and came and did a little bit extra sampling for them there, and they're very kind to allow me to sample these wells. And recently, obviously, in this year in June, Sophia invited me down with Tim when we sampled a couple of wells in Bishop's Sutton, which was great. And there's a lovely picture of that well we sampled there. The brackets are just the numbers that we found. And as you can see, one record of Aqualex, but again, that sample was right next to, in a well, right next to a river. So that probably explains that. As you see, most of the other records are Nephagus coccianus. So that's quite interesting with one Crangonyx. Whereas if I looked at all the records of the wells, then we get most of the all those species there listed. So, and the Aqualex start to dominate because that's actually driven by the Isle of Wight records. That was right. How to sample Stygofauna. Where to look? Well, back to this old sort of model, and the easiest place to look is down a well, which I describe as a hand dug thing, which is chipped out of the chalk, or a borehole, which is essentially drilled into the strata. Nelson's column's there because, just to give you an idea, that's, I think, 52 meters. Most wells are less than that. Most wells are shallow because they don't need to go very deep, so maybe 10 meters. But certainly there's one in Oxfordshire, which I know is actually deeper than Nelson's column. So that would be quite a feat to dig. Whereas boreholes can be any old depth, really. We get down to 100 meters easily in those, and those are sort of the depth that we go to the maximum of those. And the other places we can look, as we said, mines and caves, but they're difficult to get into and don't often, not in all geologies. Springs are good places to look, and then are fairly straightforward, I guess. But in a spring like that, pictured there, then we've got an issue because we'll have surface water taxa and we'll have terrestrial taxa, which will predate on the groundwater animals as they're flushed through. So, not a spring like that isn't ideal, but a spring catch pit where the spring just feeds into a tank, that's much better. Then you have the hyperharic zone, and I'm going to go on to how you sample that. That's just sort of examine a little picture, sort of trying to show what it might look like. So sampling methods. So we're going to just talk about the well net method, which is the common method. And these are sort of range of nets. You stick a weight on the end, attach it to a cable, and lower it down the net and disturb the sediment at the bottom, bring it back. Another way is what's called the Boo Rouge pump, which we use in a hyperharic zone to sample that, or the Karaman Chapuis method, which dates back to sort of early 1900s, I think, 1930s perhaps, and then different types of nets. Here is me. There's a little video of me sampling a borehole. So it's a typical environmentally borehole. There's a net with a weight on the end. I'm attaching it to the cable. That's always important. I have lost quite a few things down the wells over time. Lower that down. There it goes. So you need a net that sort of fits there, but is not going to get stuck. So obviously the different size nets for different size boreholes. Then raise it off the base. So it gets to the bottom, raise it off the base, and then drop it all the way down again. And then you're disturbing the sediment, and then you bring it back through that sediment plume. So I sort of drop it up, raise it up, drop it down 3 times, bring it to the surface. There's a little filter on this. This is like a little plankton net, adaptive plankton net. It's a 63 micron net. So to capture the fauna, and it also capture copepods and ostracods. and then I've just wash it out and then stick it back down. And then when I've done it again, and wash the whole net out properly. So what that looks like, this is from a well. The net's too small to be honest, but this is from a well in Berkhamsted. I think I did some years ago. This I'm just trying to show what it looks like. So disturbing the sediment, then you just pull the net back up. So that's that. And then this one, hopefully you might be able to see some little white dots here moving around. So those are the groundwater fauna, little stygobites crawling on this piece of metal, which turned out to be an old ordnance. So that was, yeah, that was a little bit hairy after I found that out. Anyway, so that's how it's very simple method of how you can sample a borehole. Okay, the next one is the hyperaric zone. So we're using the Buru. So as you can see, this is a pipe, very simple, with perforated holes down here, an old style pump. So you bash this into the river. You won't get that down to about 30 centimeters or 50 centimeters ish into the hyperaric zone, depending on the geology. The chalk is about 30, about 30 centimeters. And then we stick this net on, stick this top on, and then we pour water in there to prime it and then pump it. So this is what happens. General rule of thumb is to pump 5 litres of water, which you then pass through the net. It's easier to pump it into a bucket and then put it in a net. Just about doable with one person. So that's on the River Lambourne some time ago, where we did some research into what the hyperaric community was before and after restoration. So there are other methods which are less disturbing in some ways, which is a vacuum pump, a standpipe. They're all similar type things. You can sort of put that in, and then something called a Williams core, which I've never seen actually, but you could see how it operates. You take a distinct sort of sample from the base. The other methods, this is called the Karaman Shapui, but really it's just digging a hole in the ground. Mainly for sort of those sort of big alpine rivers, or you might in Scotland, it would be useful, those massive gravel bars. That's where you would use this method. Not so good on the chalk really. And you just fish that out and put that through a sieve or a net. So that Anthrobathanera has been found using this method. I mentioned spring catch pits. This is a spring catch pit, so there's a flow into a vessel where you can sample that and just use a pond net to sample it's a bit tricky, but you've got to sweep it through and disturb the sediment on the bottom. That can be quite successful. Otherwise, stick a net, let it drain through like this, or this is from an outfall from a cave, so you could put a drift net under that. And similarly, if you were in a cave, you can just go and do a regular kick sample. So what's the EA doing about groundwater ecology? So I'm just going to tell you really very briefly why we're doing it and what we're doing. So a little while ago, back in 2025, oh no, rubbish, not 2025, in 2018, the UK government published this 25-year plan and sets out sort of actions to improve the natural environment and to deliver cleaner waters and where possible to prevent sort of human-induced extinctions or loss of known threatened species in England. And these sort of are translated then into these sort of plans are translated to an environmental improvement plan, which has legally binding targets to halt the decline in species abundance and reduce that species risk of extinction. And you can see that's the sort of apex goal is to thriving plants and wildlife. And while groundwater ecology doesn't really mention in any of this, you can see it does fit in there because it's obviously a unique set of fauna which need protecting. And we need to monitor and know what status they're at. So the vehicle for delivering this plan is the Natural Capital Ecosystem Assessment Programme, which is a DEFRA-wide programme and involves organisations such as the Environment Agency, Natural England, Forestry Commission, and probably some other ones I can't remember. But you can imagine the Environment Agency is involved or tasked, I should say, with delivering this programme for the water environment. For groundwater, you can imagine there's programs for groundwater quantity, groundwater quality. There's also new programs for groundwater, so they were existing, but we've also got programs for groundwater temperature and groundwater wetlands or groundwater fed wetlands. So there's monitoring all associated with those and re-evaluating how we've done some of those other stuff in the past. This now turns our attention to groundwater, so the groundwater ecology and my design or network design, and I leaned heavily on, or took a hydrological unit approach, which is a bit of research which was developed at Roehampton University back in and published in 2017, and essentially uses 4 different rock types. I know I mentioned 3 earlier, but there's another one which is this mixed, which is sort of sandstones and other bits and bobs, and looks at those and then classify or groups those, I should say, according to their hydrogeological and hydrochemical sort of characteristics. So we've got these 11 geohabitats you can see listed on the bottom. And then the next part of that was to look at the habitat quality for stygobites. And one of the sort of drivers of that was the transmissivity of those rocks, i.e. the speed and which water can move through those rocks. And unsurprisingly, it can move through the chalk really well because it's got some massive fissures, etc. And similarly with limestones, and they was ranked according on that basis from high to low. Now, as you'll see, I've chopped off Wales for starters because Environment Agency only now covers England, but the principle applies. However, you can see most of England is actually fairly low quality, which is a bit disappointing. But I think it just shows, actually, as we know earlier on, we said that the Nephagus glenii is very much is an endemic species known well throughout Devon and Cornwall. So actually, why is that not high? So I think that needs to be addressed in some way. And so I have tried to address that in my sampling program by weighting our sampling in the favour of the area coverage and also the habitat quality. So high habitat quality or large area means you'll get more boreholes or more sites located in those things. Now, finding the sites is a lot harder than it seems. For 2024, I'd selected 55 sites, which are those green dots there on the little map there, and then next year would be another 25-ish sites, so 80 sites in all-ish will be sampling, and that might grow in time. Just to tell you about the implementation so far. So basically, I've selected boreholes mainly on less than 100 meters. Over 100 meters is possible, but it just gets a little awkward. And also, generally, you find the deeper you go, the less the dissolved oxygen. So, yeah, probably less suitable habitat. Then boreholes, the borehole diameter. So there's different size nets. I've tried to well, I have to go down to really small nets. So I have to go down to 5 centimeter or slightly, 5 centimeter net. So a borehole with slightly bigger than 5 centimeters is doable. Then look at unconfined strata because we don't want to be sampling. If it's confined, it's got an overlying layer, then the likelihood is that the food and also the dissolved oxygen will be too low. And then existing monitoring, so these are existing EA monitoring sites. Don't actually think that one in the middle is, but let's not worry about that. Then what we've done this year, we've actually gone and sampled those 55 sites. We sampled them in March this year. Before that, I trained staff in the sampling method. So we have samplers who go out and do this sort of thing. And then I've also trained some of our staff, our ecology teams to identify these animals. And yes, using that standard method, we've also collected water chemistry, so pH, temperature, nitrate, electrical conductivity, dissolved oxygen, and dissolved organic carbon. And then water level data as well. Going forward, we're going to continue with our well net surveys and fill some gaps in where we've not been able to perhaps to get to all those different geohabitats. But we also want to look at the wider network because we're not just looking at a borehole, and that's perhaps unrepresentative of the groundwater proper. Certainly unrepresentative from a microbial point of view, and no doubt the stygofauna point of view as well. So we're looking at using environmental DNA, eDNA. We'll need to pump these boreholes for that purpose. Now that's going to be a whole another set of sites that I'll need to establish, and we do some pumping, so we should be able to do that. And we've got a research project with Centre for Ecology and Hydrology and British Geological Survey doing some work on that whole aspect at the moment. And another aspect is the microbial functioning. So this is basically using a water sample and then looking at the density and activity of microbes within the sample, so a pump sample, and pushing them through this flow cytometer, which will tell us that say that density, and from that and that activity, and from that we could potentially say where we've got reference sites where we've got low activity, low density, which should mean pristine type, or pristine inverted commas, conditions, which we can then compare and contrast other sites in the same geology or same type of habitat to that sort of reference point, which is what we do in rivers, and therefore see if there's a difference. And that's hopefully where we'll go to with the microbial community stuff as well. But that's all early days, really. In terms of that groundwater, what we want to do is to be able to report on groundwater health and condition from an ecosystem point of view, then all these factors will come together to help us deliver that. So, right, identifying these things, not that difficult really, but because they're small, it becomes difficult. Here's a picture out of the FBA, Freshwater Biology Association key to Crustacea, and this is a typified amphipod, so basically Gammarus really. But the key things to consider, or not worry about, but think about, look at, are these what's called the hands, the gnathopods, the silent G. So they're really important. That's a good starting diagnostic feature. The second bit is these little things called the epimeral plates. The shape of these is important. Then we turn our attention to this little turny thing, which is hidden, which is hard to find and even harder to see sometimes, which is the mandible palps, and its sort of arrangement of hairs on that's important. And then we have the urosome and these sort of what are called the endopods and exopods, but also confusingly called rami. So these these things are important, and the shape of this little thing called the telson is also important. So those are the key issues. This is going off a slight tangent. It's useful to be able to sex them, just because there's some differences between the males and females when you're looking at them. This isn't a groundwater animal. This is Dicogamrosema bafis, which is an invasive shrimp, the demon shrimp, in fact. And this basically is the female stygites, which are coloured green, and they can easily be confused with the gills. But that's what they are. So they sort of hold the eggs in there, and then you've got the male genital papillae here, which are coloured purple. And then you can find these. You found loads of these intersexed ones, and this happens a lot in Crustacea because I think it's an invasion strategy potentially. So they end up being sort of feminized. I think that's where it is, and it's by a parasite. But anyway, so back to the amphipods, and we've got our Niphagus aquilex, Colchialis, etc. And you can see these are these different hands, and it's basically the shape of these. Are they square? Are they rectangular? Are they sort of vaguely triangular, or are they elongated? And what's this angle like? I always struggle a little bit with that, but that's that angles can be important too. And then this is the sort of arm bit, the carpus, and you can see for the female male, there's a difference in this in Niphagus coccianus. Basically, the big easy win, as I said earlier, is Niphagus fontana. It's a fairly large amphipod to start with, and think, oh, good, it's large. And then you look at these gnathopods, and one is distinctly bigger than the other, so that's a clear sign. And then Niphagus coccianus, they're square, rectangular. They're not like the other ones, so that's quite fairly easy. And again, this longer carpus. Of the other ones I just included, an easy win is these Crangonis subterraneus. They're really weirdly elongated, and so are the Micronophagus. But remember, Micronophagus is tiny and also has this weird, okay, you like the word weird, but it has this protuberance, that's the word, and then napped means it's sort of dotted. So that's quite interesting. So you find that on the Micronophagus, which can be seen only obviously under a microscope. And you can see that's sort of what the Niphagus aquilex looks like. You can definitely see this difference here, I hope, with the Fontanus. The sort of rectangular shape of these is for the Coccianus, and then these elongated ones for the Subterraneous and the Micronophagus. Glenii, sort of similar, but yeah, so different. Another easy part for Nephagus aquilex is this part, the epimeral plates. So if you find the epimeral plates are rounded, like this, like I think they look a bit like saddles, then you're onto a winner, because that's Nephagus, and that's nicely demonstrated here, Nephagus aquilex. End of story. None of the others are rounded like that, and they're all a little bit more pointy. And the Nephagus cockyi is particularly pointy. These pictures, you can sort of see that there, hopefully. The next part is the mandible palps. The Nephagus glenii is distinctive in the sense that it doesn't have this arrangement of setae on this. They called it articles. This is article 1, 2, 3, and article 3 has these D setae, which is important. And now, interestingly, they don't have it, or they have no sort of fringe there. So if you can't find it, then that's good. Hopefully, Glenn. Also, you know, it was, if you were looking at an animal which you'd found in a chalky around in Hampshire, then it wouldn't be Nephagus glenii because it's not present. Aquilex, though, has got this what's called an extensive arrangement of D setae, and this is cockyi which are apparently less extensive, although it looks quite extensive to me, but it's a little bit more sparse. And then on Micronephagus, you've got this sort of fine brush, but to be honest, you'd have worked this one out without this probably, because it would be so small to look at. The telson is really useful for Micronephagus. It's V-shaped, and you only got these little one setae off the end there. And if you're confusing Micronephagus with Crangonids because they've got similar gnathopods, these bits are very dissimilar. And then cockyi really the only one which is U-shaped. The other ones here are sort of more V-shaped, and those I guess you could are less diagnostic. But this arrangement of a number of spines and CT is quite useful. Next is the Europods. Now, I've sort of gone in the order of things I like to look at. I don't really like looking at these bits because I get a bit confused because it looks simple on the map, on the diagram, but less simple when you're looking at down the microscope. But basically, we're looking for these inner or outer rami or endopods, exopods. For Nephagus glenii, the important thing to remember for them is that this outer ramus or exopod is shorter than the inner one. So that's another useful diagnostic feature. Then Cocceianus has got this little spine, and on Aquilex, particularly this is a male one, you've got these monstrous long Europods here, these articles rather, on the Europod 1. So 3, sorry, 3. So that's quite obvious as well. But you'll see the saddle and all the other stuff, so you'd know it was Aquilex anyway, probably. But it's good to double check. You don't want to go on just one feature. And finally, the Crangonids is this bit's really, really tiny. And then what I've done, if you were going down this, interested in any of this, I've sort of written down the sizes and the sort of key diagnostic features. So hopefully that was of some use. Okay, so turning attention to the last two species, the Procellus is you're not really going to confuse it with much, as you could confuse it actually with a springtail, a collembola, which is it's not a groundwater animal, it's a terrestrial animal. It's often found, so it's often troglodytic in this. It lives underground, but shouldn't be in groundwater, but it does fall into groundwater. So you find it in groundwater a lot, and some of them are eyeless, and they look a little bit similar, but they're not segmented like this. And whilst they got a couple of prongs out the back, they're not in the same way, and they certainly don't have antennae, long antennae. So you can, of course, find Acellus aquaticus, that's what I was trying to say, or Meridionus, or Communis, I think, and they could all be confused with this. If they've lost their pigment, they may have fallen in and become underground. They may lose their eyes, but you still see some sort of residual eye, vestigial eye. And I should also mention that you can find Gammarus in the hyperaric zone, for example, or even in caves, and you think, oh, I found something, but even though they've lost their color, you still see some vestigial eye development. So that's how you would particularly distinguish those, and they don't match the other diagnostic features. I won't go into all this because this is really quite difficult, so we'll skip over that. And then finally, we've got the Syncarid, which is a superfamily, of this thing, the Anthropathenella stammeri. So very strange looking animal, has this sort of elongated body, and these number of trunk segments to the trunk and within the thorax and the abdomen. And then it has this sort of fat head, and then it's got these branch sort of things coming off these thoracic segments, apart from the, as it said there, apart from the exception of the last one. Again, you could confuse the tiddly things like this with Symphyla and other sort of subterranean animals, but they are not groundwater animals again. Like let's say like Colembla, but there's some other ones that you could find and think, oh, what's that? So it could look a little bit similar to that. Finally, how I go about sorting them. So basically, we're collecting samples. Collect samples can be like this or this, and all sorts of sizes, and then stick them through a series of sieves, and then go through those sieves. So the 63 micron sieve is the mesh is the smallest sieve that I use. I use a small petri dish. This thing can be used, but this I don't recommend it. And this is a small petri dish with little grates on it, so you can go through and work out as you put that under the microscope. And you sort of light from one side is helpful to sort of identify things. So that's what I'm using. I'm very fortunate to have a nice microscope in which to do it, but it is possible on a bog standard normal sort of microscope. This isn't too difficult, I'd say, to separate those sort of 6 amphipods particularly, and the process as well. Right, so this is my final slide. So this is the further information. There's Hypergene Crustacean Recording Scheme. That's a link to that. That you can get the database from that. You can also get this little thing called the Cave Life in Britain, which is published by the FBA, but it's by Lee Knight. So that's quite a useful little PDF. Hyper-egg sampling, if you wanted to dig a hole and do that Karaman-Shapui or bash something into the river. This is a SEN guidance which I was involved in for rivers. There's this Environment Agency review of subterranean aquatic fauna, which I managed in 2009-10, and then a paper which I did for sort of the predecessor to that geohabitat work. And that's it really. I'm sure there's some other thing I could link to. So yes, thank you very much.